Our history is not just written in books. It is etched into our very bodies, waiting to be read. For the vast expanse of human time, there were no chronicles or diaries to record the epic journeys of our ancestors. Entire populations moved across continents in a silence that seems absolute, yet they left behind a silent archive, one we are only now learning to decipher. This archive is made of teeth and bones. Modern science has become a master decoder, extracting incredible stories of movement, diet, and kinship from the chemistry and genetics preserved within these ancient remains. By the end of this exploration, you will see how these biological clues are completely rewriting the story of our shared past. We will uncover how a single tooth can reveal a childhood homeland thousands of miles away, and how DNA from a skeleton can trace a migration that toppled empires. How much of what we think we know about ancient peoples is about to change? Think of a tooth as a tiny, indestructible diary. The enamel that coats it is the hardest substance the human body produces, a crystalline fortress that seals a moment in time. From the moment a tooth forms in childhood until the day it is lost, its chemical composition remains locked in place, untouched by the passing years. This enamel captures a snapshot of the water you drank and the food you ate during those early years, a permanent record of your geographic origins. Bones, in contrast, are living archives. They constantly renew themselves, absorbing elements from your environment throughout your life. This means a single skeleton can tell two stories: the story of where you began and the story of where you ended your journey. These biological materials offer something profoundly rare, a direct, unedited data stream from the past. For 99% of human history, there were no scrolls, no carvings, no written word to mark a people's passage. Pottery shatters, textiles decay, and wood returns to earth, but a molar buried for millennia remains. It is a resilient witness, and when we examine not just one set of remains but many, these individual stories begin to merge into something far greater. They become the collective biography of a people, revealing patterns of movement, connection, and survival that no single artifact ever could. This is the silent archive, a library built not of paper but of biology, waiting for us to learn its language. The language of this silent archive is written in chemistry. To read it, scientists turn to a method called stable isotope analysis, a way of measuring subtle variations in the building blocks of life. Everything we eat and drink, from a handful of berries to a cup of water, contains atoms of elements like carbon and nitrogen. These atoms come in slightly different forms, called isotopes, some heavier and some lighter. The key is that plants and animals from different environments collect these isotopes in unique ratios. A person living by the sea, eating fish and shellfish, will have a very different isotopic signature in their bones than someone living on the plains, eating wild grains and the meat of grazing animals. Their bodies literally incorporate the landscape into their very structure. So when researchers extract collagen from ancient bone or analyze the pristine enamel of a tooth, they are not just looking at chemistry. They are reading a lifelong menu, a direct record of what that individual consumed. For instance, the spread of maize agriculture across the Americas left a clear isotopic trail in human remains. A society that adopted farming corn has a distinct carbon signature compared to their foraging neighbors. This dietary fingerprint becomes a powerful, if indirect, clue to geography. If a skeleton discovered in one region carries the isotopic signature of a diet from hundreds of miles away, it tells a clear story. That person or their parents before them made a journey. Their bones remember a homeland their mind may have forgotten. But the most precise tracker of movement lies in the very bedrock beneath our feet. Strontium is a fascinating element that acts like a geological GPS system encoded into human remains. As rainwater filters through the ground, it dissolves tiny amounts of the local rock, picking up a unique blend of strontium isotopes. This signature enters the soil, is absorbed by plants, and travels up the food chain, eventually becoming part of your body as you eat and drink. The crucial detail is that tooth enamel, forming only in childhood, captures and permanently locks in the strontium signature of wherever you lived during those early years. Bone, constantly remodeling, reflects where you lived in the later chapters of your life. This creates a powerful forensic comparison. If the strontium in an individual's tooth enamel does not match the strontium in their bones, or the geology of their final resting place, it tells an undeniable story of a life lived across distances. Imagine an individual laid to rest in a lush river valley. Their bones show the strontium of that fertile soil, but their teeth tell a different tale, carrying the signature of an ancient granite mountain range hundreds of miles away. This person did not simply travel as an adult. They were born in one world and died in another. Their journey, unrecorded in any text, is written plainly in the chemistry of their skeleton, a silent testament to a life of profound movement. The water you drank as a child tells its own story, written in a different chemical dialect. This brings us to the third key system, oxygen isotopes, which act as a natural recorder of ancient climate and landscape. Rainwater does not fall with a uniform chemical signature. As clouds move across continents, shedding their moisture, the heavier oxygen isotopes tend to fall first. This means that rainfall becomes progressively lighter in its oxygen isotope composition the farther inland you travel, and the higher in altitude you go. Coastal regions have one signature, mountainous interiors have another. Every sip of water from a village well or a rushing stream carries this distinct climatic fingerprint. And just like strontium, this oxygen signature is permanently captured in the enamel of your teeth as they form. So while strontium points to the underlying geology of your childhood, oxygen reveals the climate and hydrology of that same place. It tells scientists whether you grew up in a wet, coastal environment or a dry, continental interior, high in the mountains or down on the plains. On its own, this is a powerful clue. But the real investigative magic happens when you combine the maps. Think of strontium as pinpointing a specific type of rock on a continental map. Oxygen then overlays a climate map on top of it. The intersection of those two signals, the unique geological and climatic address locked in a single tooth, can narrow down a possible origin to a remarkably specific region. For an individual found far from home, this dual signature becomes a compelling coordinate. It can distinguish between someone who moved from one river valley to the next, versus someone who undertook a monumental journey from a humid coastal homeland across a mountain range to an arid desert basin. Their body carried a map they never knew they had, a map we are only now learning to read. But chemistry, for all its precision, can only tell us where someone lived and what they ate. It cannot tell us who they were related to or the deeper biological legacy they carried with them. For that, we must move from the chemistry of the landscape to the very blueprint of life itself, the genetic code. The last 2 decades have seen a revolution in our ability to recover and read ancient DNA from skeletal remains. This is not the DNA from a living person, but fragile, fragmented genetic material preserved for centuries or millennia, a whisper from the past that modern technology has finally learned to amplify and understand. This ancient DNA allows researchers to do something astonishing, to trace direct biological kinship across vast stretches of time. By comparing the genetic profiles of individuals buried together, scientists can identify family groups, parents and children, or cousins laid to rest side by side. A burial mound is no longer just a collection of skeletons. It becomes a family portrait, revealing the social structure of a community through their shared genetics. On a grander scale, this genetic data reveals the deep history of entire populations. It can show when two genetically distinct groups met and mixed, their descendants carrying the legacy of both. More dramatically, it can reveal instances where one population seems to have completely replaced another. A story of migration and movement written not in pottery styles or tool types, but in the fundamental biological instructions of the people themselves. Where isotopes trace an individual's journey, ancient DNA maps the journey of their bloodline, revealing chapters of human history that left no other trace. For decades, one story dominated our understanding of how humans first arrived in the Americas. It was a theory built on stone, quite literally. The discovery of distinctive, finely crafted spear points, known as Clovis points, across North America suggested a single, rapid migration. According to this model, a group of highly skilled hunters crossed the Bering Land Bridge from Siberia around 13,000 years ago as the last ice age retreated, and then swept southward in a wave, their signature tools marking their path. This Clovis first narrative was clean, compelling, and for a long time it was the textbook answer. But science, when faced with new evidence, must always be ready to rewrite the textbook. That evidence came not from stone tools but from bones, the remains of a young individual found in Montana and dating to nearly 12,700 years ago provided a pivotal clue. This individual, often referred to as the Anzac child, was buried with Clovis artifacts, directly linking the culture to a real person. When scientists successfully sequenced this ancient DNA, the genetic story was clear and profound. The child's genome showed an undeniable and close relationship to modern Native American populations, and crucially, to ancient peoples of Siberia. This was the long-sought biological proof of the Beringian origin. Yet, the genetic data also contained a twist. The depth of the divergence suggested these ancestral populations had been isolated, likely on the Bering Land Bridge itself, for thousands of years before moving into the Americas. This meant the journey was not a single late dash, but a long, patient saga of survival in a frigid, changing landscape, pushing the timeline of the first arrivals back even further than the Clovis era. The stones told part of the story, but the bones revealed the deeper, more complex biography of the people who carried them. As these pioneering groups finally moved south, fanning out across two vast continents, their later histories are written in the chemistry of their remains. Isotopic studies on skeletons from thousands of years later show incredible diversification. Some groups show signatures of a lifelong reliance on maritime resources along the Pacific coast, while others, deep in the interior, carry the clear mark of a land-based diet, tracking the great herds of the plains. Each set of remains adds a sentence to this epic chapter, tracing how one founding population gave rise to the astonishing tapestry of cultures that would eventually call the Americas home. While the Americas tell a story of deep ancestral roots, another continent witnessed a transformation so profound it reshaped humanity's relationship with the Earth itself. This is the Neolithic Revolution, the dawn of agriculture, and its arrival in Europe stands as one of history's great puzzles. For generations, scholars debated a fundamental question. Did this revolutionary knowledge, the practice of sowing seeds and herding animals, spread as an idea? Did local hunter-gatherer communities see their neighbors farming and simply adopt the practice, a process called cultural diffusion? Or did this new way of life arrive with new people, carried by migrating farmers who moved across the landscape, bringing their crops, animals, and traditions with them? The debate was fierce, with arguments based on the spread of pottery styles, tool technologies, and settlement patterns, but the answer, as it so often is, was waiting in the bones. When scientists began extracting ancient DNA from skeletal remains found across Europe, from the earliest farming sites, they uncovered a genetic story that settled the debate with stunning clarity. The data revealed a massive wave of migration, a movement of people originating from the region of Anatolia in modern-day Turkey. As these early farmers moved westward across Europe over generations, their genetic signature spread with them. In many areas, the local hunter-gatherer populations did not simply adopt farming. They were largely replaced by the incoming groups. The genetic evidence shows only limited mixing in most regions, painting a picture of a continent undergoing a dramatic demographic shift. This was not merely the spread of a technology, it was the movement of entire communities, families, and ways of life, written indelibly into the European gene pool. Yet the story does not end with the broad genetic map. Isotope analysis on these very same farmer skeletons adds an intimate, human scale to this epic migration. We can now examine individuals from a farming village in Central Europe and ask a personal question. Where were you born? The strontium and oxygen isotopes locked in their teeth often tell a surprising story. Many show a childhood signature that does not match the local geology of their adult burial site. Instead, it points eastward toward the direction of Anatolia. This means a significant number of these early farmers were not born in the villages where they died. They were migrants or the children of migrants who carried their agricultural tradition with them on a lifelong journey. Their bones hold the chemical record of that trek, a personal itinerary within the grand historical narrative. One individual's isotopes might show they spent their childhood several hundred kilometers to the east, while another, buried in the same community, might show a local signature, perhaps the child of the first generation to truly put down roots in that new land. Together, the DNA and the isotopes create a multi-layered history. The genetics show the population-level replacement, the grand tide of movement. The isotopes reveal the individual voyages within that tide, the lived experience of families pulling up stakes, traveling across unfamiliar landscapes, and founding new homes in a changing world. It transforms the Neolithic Revolution from an abstract historical event into a saga of countless personal decisions, hardships, and hopes, each one leaving a tiny, chemical signature for us to find millennia later. The story of post-Roman Britain presents a different kind of puzzle, one where written history and physical evidence engage in a fascinating conversation. After the last Roman legions departed in the early 5th century, the historical record, penned by later chroniclers like the Venerable Bede, tells of a great invasion. Waves of Angles, Saxons, and Jutes, Germanic peoples from across the North Sea, arrived in their ships, battling the native Britons and establishing new kingdoms. For centuries, this was accepted as a story of conquest and replacement, where the old Celtic and Romanized British culture was swept aside by a new Germanic order. It was a narrative of clear-cut change, reflected in new languages, new burial practices, and new forms of governance. But how many people actually came? Was this a massive migration of entire communities, or a smaller military takeover by a ruling elite? The bones in the ground held a more nuanced truth. Archaeologists turned to a key site, a large early Anglo-Saxon cemetery at West Heslington in northern England. Here, they applied the same isotopic detective work used on Neolithic farmers. They analyzed the strontium and oxygen isotopes in the teeth of individuals buried in the 6th and 7th centuries. The results were striking. A significant portion of the people, both men and women, showed isotopic signatures that were inconsistent with the local chalk geology of Britain. Their childhood chemistry pointed unequivocally to a homeland across the North Sea, in regions like modern-day Denmark, northern Germany, and the Netherlands. This was powerful physical proof that the historical accounts were correct in one vital aspect. A substantial number of migrants did indeed arrive from the continent. These were not just warriors, but whole families, women and children included, who made the perilous sea crossing to build a new life. Their teeth confirmed the journey that history books had described. Yet, when geneticists began to extract ancient DNA from these same early medieval cemeteries, they introduced a compelling complication. The genetic data revealed a story not of simple replacement, but of profound integration. While the Anglo-Saxon migrants did contribute substantially to the gene pool of what would become England, the majority of the genetic ancestry in early medieval individuals came from the existing pre-migration population of Britain. The local Britons were not wiped out or entirely displaced. Instead, over generations, they adopted the new language, the new material culture, and the new social structures of the incoming groups, blending with them. This genetic picture transforms the narrative from one of invasion to one of cultural fusion and gradual change. The old idea of a clean ethnic break between Romano-British and Anglo-Saxon England dissolves. So how do we reconcile the clear isotopic evidence of migration with the genetic evidence of continuity? The answer lies in understanding the nature of power and cultural change. A smaller but socially dominant group of migrants can effect a dramatic cultural shift without requiring a total population replacement. The Anglo-Saxon language, laws, and customs became the new standard, the path to status and influence. Over time, through intermarriage and social assimilation, the genetic lines blurred even as the cultural landscape was remade. The physical evidence gives us both sides of the same coin. The isotopes capture the moment of movement, the personal stories of individuals who crossed the sea. The DNA reveals the long-term outcome, the weaving together of two peoples into one. It shows that history is rarely a story of pure conquest or pure continuity, but a complex tapestry of movement, meeting, and mixture. This leaves us with a profound question for other historical transitions, where the written record is silent or one-sided. How many other foundational stories of peoples and nations, stories we accept as simple truths, might be waiting for their silent archives to speak? Around the year 1200 before the Common Era, the civilized world seemed to come apart at the seams. From the palaces of Mycenaean Greece to the great Hittite Empire of Anatolia and the wealthy city-states of the Levant, a wave of destruction rolled across the eastern Mediterranean. Grand cities were burned and abandoned, trade routes linking continents fell silent, and complex writing systems were forgotten. This was the Bronze Age collapse, a catastrophic unraveling that plunged the region into a centuries-long dark age. Among the few written records that survive from this chaotic time, one name echoes with particular menace: the Sea Peoples. Egyptian inscriptions on temple walls at Medinet Habu describe a terrifying coalition of foreign invaders who attacked by land and sea, a confederation of groups with names like the Peliset, the Shekelesh, and the Denyen. The pharaoh Ramesses III claims to have defeated them in a great battle, saving Egypt from the fate that befell its neighbors. But who were these mysterious marauders? For centuries, they have been ghosts in the historical record, blamed for the collapse but leaving no clear homeland, no definitive archaeology of their own. Were they a cause of the collapse or a symptom of it, desperate refugees fleeing a wider systemic failure? The texts give us names and dramatic reliefs of battles, but they offer no physical evidence, no bones to put to the names. That evidence, however, may finally be emerging from the ashes of the very cities they supposedly destroyed. At ancient sites like Tel Es Safi, identified as the Philistine city of Gath, archaeologists are uncovering a grim but informative record. They find layers of intense conflagration, and within them the remains of individuals who met a violent end. These are not orderly burials, but the tragic casualties of a city's final hours. By applying isotopic analysis to these skeletons, scientists are asking a direct and poignant question. Among the dead, can we identify those who were not from here? If the Sea Peoples were a real coalition of foreign fighters, then their remains, fallen in a destroyed city, should carry the chemical signatures of distant homelands. Early studies are revealing exactly that. Some individuals show strontium and oxygen isotopes inconsistent with the local environment, pointing to origins across the Mediterranean, perhaps from the Aegean or Anatolian coasts. This does not mean every foreign skeleton was a sea people's warrior. The period was one of immense upheaval and displacement, but finding groups of young men with non-local signatures among the ruins of a sacked city provides the first tangible, physical evidence that aligns with the Egyptian texts. It suggests that among the chaos of the collapse, there were indeed organized groups of outsiders moving with force, their journeys now etched not just in stone carvings, but in the chemistry of their bones. This transforms the sea peoples from a mythic explanation into a historical population we can begin to study directly, understanding their movements and their role in one of history's most profound resets. Across the African continent, a different kind of migration unfolded, one so vast it reshaped the cultural and genetic landscape of an entire subcontinent. This is the story of the Bantu expansions, a millennia-long movement of peoples that began in the region of modern-day Cameroon and Nigeria and spread south and east, eventually reaching the southern tip of Africa. For a long time, our understanding of this epic journey came from two indirect sources: the spread of a distinctive pottery style and, most tellingly, the distribution of over 500 related languages that form the Bantu family, spoken by hundreds of millions today. The archaeological trail showed a clear pattern, moving with the advance of Iron Age technology and agriculture, but a critical question remained. Did this represent a true migration of people, or was it again the spread of ideas, with local communities adopting new technologies and languages from neighboring groups? The answer, hidden in ancient burials, reveals a powerful story of movement written in biology. Early studies focused on a subtle but telling clue: dental morphology. Researchers noticed that the shape and specific traits of teeth in skeletal remains from early Bantu-associated sites in southern Africa were statistically distinct from those of the indigenous foraging communities, like the San people. This biological signature suggested the arrival of a new population, not just new parts or words, but the most definitive evidence has come from the field of ancient DNA, a tool that is finally beginning to illuminate the deep past of Africa. Recent studies sequencing DNA from skeletons in South Africa, dating to the Iron Age, have found a crucial link. These individuals show a clear genetic connection to populations in West Africa, the very cradle of the Bantu expansions. This genetic data provides the direct biological confirmation that this was a story of demic diffusion, a movement of people. Families, farmers, and ironworkers carried their language, their culture, and their genes across thousands of miles over generations. Their journey was not a sudden invasion, but a gradual, relentless expansion, often following river valleys and adapting to new environments. The silent archive of their bones and teeth now corroborates the story told by their pottery and their words, weaving together linguistics, archaeology, and genetics into a single, coherent narrative of one of humanity's most significant, yet least textually recorded chapters. The glittering palaces of the Aegean hold their own genetic secrets. On the island of Crete, the sophisticated Minoan civilization flourished, famed for its vibrant frescoes and labyrinthine palaces. On the mainland, the fortified citadels of the Mycenaeans arose, their rulers buried in golden masks. For over a century, archaeologists have debated their origins. Were these the creations of indigenous peoples, or did their founders arrive from across the sea, perhaps from Anatolia or even Egypt? The myths spoke of migrations, but the physical evidence was silent until we learned to ask the bones themselves. By extracting ancient DNA from the remains of individuals buried in the heart of these civilizations, scientists have written a new origin story. The genetic data reveals a profound and shared foundation. Both the Minoans of Crete and the Mycenaeans of mainland Greece were overwhelmingly descended from the same early Neolithic farmers who had migrated from Anatolia thousands of years earlier. Their primary ancestry was not from some distant, mythical homeland, but from the deep agricultural roots of the region itself. This genetic continuity suggests that these dazzling Bronze Age cultures were, at their core, a magnificent local development, the flowering of a population that had been in the Aegean for millennia. Yet the story diverges when we look north. The Mycenaean genomes contained a crucial extra ingredient, a significant genetic component linked to the pastoralist peoples of the Eurasian steppe. This steppe-related ancestry was largely absent from the Minoans. This finding paints a vivid picture of later prehistory. It suggests that after the initial settlement by Anatolian farmers, a second major pulse of migration reached mainland Greece from the north, bringing with it not only new genes but likely new cultural elements, perhaps even an early form of the Greek language. The Mycenaeans, then, represent a fusion, the meeting of the ancient Aegean world with influences from the vast continental interior. Their famed warrior culture and their role as the heroes of Homer's epics may find a distant echo in this genetic signature from the steppe. Meanwhile, the Minoans of Crete, protected by the sea, remained more genetically isolated, a unique branch of the Aegean family tree. So the silent archive clarifies a long-standing mystery. It tells us these were not foreign implants, but homegrown marvels whose distinct characters were shaped by different chapters of ancient migration, one deep and foundational, the other more recent and transformative, both written in the code of their bones. Across the vast, empty expanse of the Pacific Ocean, another great migration unfolded, not on horseback or foot, but in outrigger canoes. This is the story of the Lapita people, the first explorers to push beyond the visible horizon into the remote islands of Oceania. Their journey is one of humanity's most audacious chapters, a purposeful expansion across thousands of miles of open sea, and it began around 3,500 years ago. We know them primarily by their calling card, a stunningly distinctive style of pottery decorated with intricate, stamped patterns. Where this pottery appears, from the islands north of New Guinea all the way east to Samoa, it maps a trail of settlement across the Pacific. But pottery alone cannot tell us who these people were or the intimate details of their voyages. The tropical climates of these islands are unforgiving archives, where heat and humidity rapidly break down organic material, including human bone. For a long time, the Lapita people were ghosts defined by their artifacts, their physical presence lost to time. Finding their actual remains, the biological proof of their journey, became one of archaeology's great quests. The breakthrough came from rare, preserved cemeteries on islands like Vanuatu and Fiji. Here, in these ancient resting places, scientists finally recovered something precious: Lapita teeth. While bone may decay, dental enamel endures, and within these tiny time capsules, the chemical story of a lifetime at sea was waiting. Isotope analysis of the teeth revealed a diet heavily reliant on marine resources, but with a fascinating gradient. Individuals from earlier Lapita sites showed stronger signatures of open ocean fish, the catch of deep-sea voyagers. Those from slightly later periods on the same islands, or on islands farther along the migration chain, began to show more evidence of reef fish and terrestrial animals, like pigs and chickens they brought with them. This dietary shift, locked in the chemistry of their enamel, paints a vivid picture of the migration process itself. It suggests the first arrivals were truly oceanic people, living off the deep sea as they explored. Then, as they established stable colonies and their transported animals reproduced, their diet, and thus their isotopic signature, gradually localized. This chemical data beautifully complements the archaeological model of a rapid, purposeful island-hopping expansion. It transforms the abstract idea of a migration into a tangible story of adaptation, showing how these incredible navigators shifted from a life sustained by the open ocean to one rooted on new shores, their teeth recording each step of that culinary and cultural transition. The saga of the Vikings is written in fire and poetry, a tale of dragon ships and warrior kings. But beneath the epic poems and chronicles of raids lies a deeper, quieter story of movement, one told by the people themselves in their final resting places. From the coasts of Ireland to the remote shores of Iceland, Viking Age burials hold individuals who participated in this vast diaspora not as legendary figures, but as ordinary people. By applying our toolkit of isotopes and ancient DNA to these graves, the mythic narrative gives way to a complex human reality. Take a burial ground on the Isle of Man, a strategic hub in the Irish Sea. Isotopic analysis of teeth from several skeletons buried with Norse artifacts revealed a surprising mix. Some individuals carried the childhood chemical signature of Norway or Denmark, clear migrants from the Scandinavian homeland. Others, however, showed a local isotopic profile. These were not Viking raiders from across the sea, but local men and women who had adopted Norse culture, were buried in its fashion, and had become part of the new social fabric. This pattern repeats. In early Icelandic cemeteries, studies show a majority of the founding population were of Scandinavian origin, but a significant minority came from the British Isles, likely reflecting the taking of slaves or partners during earlier raids and expeditions. These individuals, far from home, carried the geological memory of Scotland or Ireland in their teeth, their presence silently documenting the interconnected and often harsh nature of these settlements. The genetic legacy of this period is still being calculated. Ancient DNA from Viking-era bones confirms the stories of travel and mixture, showing Norse genetic input across a wide arc from Greenland to Russia. But perhaps more telling is the living archive, the DNA of modern populations. In places like the Orkney and Shetland Islands, genetic studies reveal a striking proportion of Scandinavian ancestry in today's inhabitants, a quiet, biological echo of settlement that persisted long after the longships were gone. This data allows historians to move beyond guessing about the scale of migration. It provides a measure suggesting that in some areas, the Norse influx was substantial enough to leave a permanent demographic imprint, while in others, like mainland England, their genetic impact was more limited, a wave that receded after leaving a profound cultural mark. The silent archive reframes the Viking Age. It was not merely an era of hit-and-run raids, but a protracted process of exploration, trade, and settlement conducted by a mosaic of people, both Scandinavian and local, whose lives and journeys are now emerging from the ground, one tooth and one bone at a time. Catastrophic events leave a different kind of fingerprint, one written not in the steady rhythm of generations, but in the sudden, stark silence of mass graves. Across Europe and the Near East, archaeologists occasionally uncover sites that speak of a single, overwhelming crisis, long trenches filled not with individual burials but with dozens laid to rest together in urgency. These plague pits, as they are often called, are the archaeological signature of pandemic disaster, from the Justinianic plague of the 6th century to the infamous Black Death of the 14th. For a long time, these sites were understood in the broadest terms as tragic evidence of a society pushed beyond its limits, but the silent archive within these pits holds far more specific stories. The first question is one of identity. What pathogen caused this? Here, ancient DNA provides a definitive answer. By extracting genetic material from the dental pulp of victims, researchers have been able to identify the unmistakable signature of Yersinia pestis, the plague bacterium, in skeletons from these mass contexts. This genetic confirmation transforms a historical suspicion into a scientific fact, linking a specific biological agent to a specific archaeological event. But the next question is more human. Who were these people? Isotopic analysis offers a haunting insight. In a crowded medieval city struck by the Black Death, you would expect the victims to be a cross-section of the local population. Yet studies of teeth from London plague pits tell a more complex story. While many individuals show the local isotopic signature of the Thames Valley, a significant number do not. Their teeth carry the chemistry of the English countryside, of distant regions, even of continental Europe. These were not lifelong Londoners. They were recent arrivals, perhaps migrants drawn to the economic opportunities of the great city, only to find themselves trapped there when the epidemic hit. Their presence in a mass grave reveals a hidden dynamic of crisis, that pandemics could freeze normal movement, trapping populations in place, while also creating a stark snapshot of the diverse, mobile world that existed just before the catastrophe. It shows how a single, terrible event could capture, in one grim sample, the very human flows of an era, preserving a moment of interconnectedness that ended in profound loss. This silent testimony forces us to reconsider these pits not just as symbols of death, but as accidental archives of life, movement, and vulnerability in the medieval world. The silence of the Tarim Basin in western China holds one of archaeology's most visually arresting mysteries. For decades, explorers and researchers have uncovered remarkably preserved mummies in the parched sands, individuals with light-colored hair, woven woolen textiles, and facial features that seemed strikingly familiar to Western observers. Dating back to around 2,000 years before the Common Era, these bodies were so intact that their tattoos were still visible on their skin. The immediate and seemingly obvious conclusion was that they represented a lost band of long-distance migrants. The prevailing theory painted a picture of hardy travelers from the Black Sea region or the Iranian plateau, who undertook an epic journey along the fabled Silk Road thousands of years before it was named, bringing their culture and their distinct appearance to the heart of Asia. It was a story that fit neatly into older ideas about ethnicity and movement, a clear westward fingerprint in an unexpected place. Then, the silent archive within the mummies themselves was asked to speak. Recent advances in ancient DNA analysis allowed scientists to extract genetic material from these very remains. The results were not just surprising, they completely overturned the narrative. The genetics revealed that these individuals were not recent migrants from the West at all. They showed no detectable ancestry from the farming populations of Anatolia or Iran that were contemporary to them. Instead, their genetic profile was almost entirely derived from an ancient North Eurasian population, a group of hunter-gatherers who had largely disappeared from other parts of the world millennia earlier. This was a profound revelation. The Tarim Basin mummies were not foreigners who had trekked across continents. They were a local, isolated remnant of one of the most ancient human lineages who had developed their unique culture, their woven clothing, their rituals, their dairy-based diet evidenced by residues in their pots in complete genetic isolation for thousands of years. Their physical appearance, which had so captivated and misled observers, was a relic of a deep, shared ancestral past, not a sign of recent travel. This finding challenges our most basic assumptions. It forces us to decouple physical appearance from cultural origin and recent migration. A person who looks one way is not necessarily from a place where everyone looks that way today. Their story is a powerful lesson in humility for historical interpretation, showing that the most obvious answer is often just a reflection of our own modern biases. The mummies were not a lost piece of Europe in Asia. They were a unique and brilliant chapter in the human story of Asia itself, a chapter written in genes, not in miles traveled. But this silent archive is not always a straightforward text to read. The stories told by bones and teeth can sometimes present puzzles, even contradictions, that remind us science is a process of interpretation, not magic. Isotopes and ancient DNA are powerful tools, but they come with their own limitations and blurry edges. An isotopic map is not a precise postal code. Many different regions can share similar geological or climatic signatures. The strontium from a chalk plain in one country might be chemically indistinguishable from a chalk plain hundreds of miles away. An oxygen signature from a rainy coastal valley could match that of many other rainy coastal valleys across a continent. The data gives us a probable region of origin, not a specific village. And sometimes the maps overlap in confusing ways, offering multiple possible homelands for a single individual. Ancient DNA faces its own challenges. Genetic material degrades over time, and samples can be contaminated by the DNA of archaeologists, museum curators, or even the soil itself. In some climates, nothing usable survives at all. This means the genetic record is full of gaps, a patchwork quilt of information where some squares are brilliantly clear and others are forever missing. These limitations become most fascinating when the two methods seem to tell different stories. Consider a hypothetical scenario built from real archaeological dilemmas. A skeleton is excavated from a burial mound in Central Europe, dating to the early Bronze Age. Isotopic analysis of the teeth reveals a signature perfectly consistent with the local geology. Everything suggests this was a person born and raised right here. But when scientists succeed in extracting ancient DNA, the story flips. The genetic profile shows a significant portion of ancestry originating from the distant Pontic-Caspian steppe, hundreds of miles to the east. How do we reconcile this? The isotopes say local, the DNA says foreign. The answer lies in thinking across generations. The isotopes capture one lifetime, the individual's own childhood. The DNA captures a lineage, the accumulated ancestry of their parents, grandparents, and beyond. This person likely had a grandparent or great-grandparent who made that long journey from the steppe. That migrant ancestor married into the local community. Their child, and then their grandchild, our individual, was born right here, drinking the local water and eating the local food, thus acquiring the local isotopic signature. Yet they still carried the genetic legacy of that ancestral voyage. This is not a contradiction, but a richer, more layered history. It forces researchers to move beyond simple labels of local or foreign, and to interpret complex biographies of integration. The bones tell two true stories simultaneously, one of a life lived in a specific place, and another of a deep family memory of movement from somewhere else entirely. These powerful tools, however, raise a profound and necessary question: whose story is being told? The silent archive we have been exploring is not made of anonymous artifacts. It is composed of people, individuals who lived, loved, and were laid to rest by their communities with purpose and care. For many, especially indigenous peoples around the world, these are not specimens, they are ancestors. The history revealed by a tooth or a femur is not abstract. It is a direct biological link to living cultures and living descendants. For decades, the field of archaeology often operated with a sense of detached scientific entitlement, collecting and studying human remains with little consultation from the communities most intimately connected to them. This created a painful legacy of disconnect, where the very bones that held a people's history were held in distant institutions, separated from their descendants. Science, in its quest for knowledge, sometimes forgot the humanity at its core, but a significant and essential shift is now underway. Across the globe, there is a growing movement toward ethical collaboration. This means that before any study begins, researchers are increasingly required to seek permission and guidance from descendant communities. It is a process built on dialogue and respect, recognizing that communities have a vital stake in how their own history is investigated and told. This collaboration has led to the repatriation of thousands of ancestors' remains to their communities for reburial, a restorative act of returning people to their homelands. For studies that do proceed, the concept of ethical sampling has emerged. This approach uses the absolute minimum amount of material necessary, sometimes just a few milligrams of powder from a tooth, to gain the required data. It treats each sample not as a disposable resource, but as a sacred, finite piece of heritage. The goal is to learn from the ancestor while causing the least possible disturbance, honoring their resting place. This ethical framework transforms the work from extraction to partnership. When a community is involved from the outset, the questions asked can change. The science is directed not just by academic curiosity, but by the community's own desire to understand its past. A genetic study might be undertaken to confirm oral histories of migration, or isotopic analysis might be used to reconnect with ancestral lands. In these cases, the silent archive becomes a bridge, not a barrier. It serves to reconnect people with a history from which they were severed, providing tangible evidence that validates deep cultural memory. The stories written in bone and tooth are powerful, but they must be told with reverence. True understanding comes not just from decoding chemistry and genetics, but from listening to the living voices connected to that past. When science proceeds with this humility, it does more than reconstruct history, it helps to heal it. A single skeleton tells a compelling story, but it is just one sentence in a much larger volume. The true power of this silent archive emerges when we move from the individual to the population, when hundreds of these personal stories are gathered and compared. Scientists do not simply collect data from one burial site. They build vast databases, compiling isotopic and genetic information from individuals across continents and millennia. It is within these aggregated patterns that the grand narratives of prehistory finally come into focus. We stop asking where one person came from and start asking how entire communities moved, interacted, and transformed. For instance, by analyzing the remains of many individuals from a single era, researchers can often detect a fascinating gendered pattern in movement. In some early farming societies, studies show that women buried in a village frequently have non-local isotopic signatures, while the men show local ones. This suggests a practice called patrilocality, where men stayed in their birth community and women moved from neighboring villages to marry. Their teeth remember the home they left behind, creating a chemical map of social networks and kinship ties that stretched across the landscape. Conversely, other patterns emerge, like evidence of long-distance trade routes that brought small, isolated groups of foreigners into a region, their distinct genetics appearing like brief flashes in the local gene pool before fading away. The scale and speed of a migration also become clear. A slow, creeping advance of a new population leaves a gradual genetic gradient. A rapid, sweeping movement looks like a stark replacement in the archaeological and genetic record. All of this requires a symphony of data, where each skeleton provides a single note, and only together do they create the melody of history. The story of the Yamnaya people from the Eurasian steppes provides a masterclass in this synthesis. For a long time, archaeologists knew these pastoralists had sophisticated wheeled vehicles and buried their elite in great mounds called kurgans, but their impact was a subject of fierce debate. Then, ancient DNA from skeletons across Europe revealed a startling genetic upheaval around 5,000 years ago. A massive influx of steppe-related ancestry appeared, dramatically altering the genetic makeup of the continent. This was the Yamnaya expansion, but DNA alone could not tell us how this happened. Was it a violent invasion, a peaceful migration, or something else? By adding isotopic data from hundreds of Yamnaya and early European skeletons, a nuanced picture developed. The isotopes showed that many Yamnaya individuals buried in new regions still carried the chemical signature of the steppe in their teeth, meaning they were first-generation migrants, not people who had been born into a blended culture. Furthermore, the data revealed that this was not just a movement of men. Whole family groups, including women and children, made the journey. This combination of evidence, the clear genetic signal, and the personal chemical testimony of movement, paints a coherent portrait of Adamic migration, a movement of people on a transformative scale. It was likely driven by a combination of factors, including climate shifts, the domestication of the horse, and the mobility offered by wheeled carts. They moved westward, integrating with and often largely replacing the existing Neolithic farming communities, introducing not only their genes but also, many linguists believe, the precursor to most of the languages spoken in Europe today. From a scatter of individual bones across the plains, a world-changing historical event was reconstructed, not from a king's decree, but from the collective memory held in the teeth and bones of the people who lived it. This silent archive does more than fill in gaps, it rewrites entire chapters. For centuries, our understanding of the past was shaped by a very particular kind of record, the chronicles of literate elites. Kings commissioned monuments to their victories, scribes recorded dynastic successions, and poets sang of epic battles. This gave us a history dominated by named individuals, dramatic events, and political borders. It was a story told from the top down, a narrative of power. But what about the farmers who tilled the soil, the families who built their homes, the generations who moved across landscapes in search of a better life? Their stories were considered lost, swallowed by time. Now, the chemistry of a tooth and the genetics of a bone are changing that. This science shifts the focus from the throne room to the village, from the general to the grandmother. It recovers the biography of the anonymous masses, the literally silent majority who built the world upon which kings stood. We are no longer just reading history, we are listening to it, one genome and one isotope ratio at a time. This shift fundamentally challenges some of our most persistent cultural myths. One of the most powerful is the idea of a pure, ancient people rooted in a specific landscape since time immemorial. National stories often begin with such an origin, a foundational population from which a modern identity flows. The silent archive reveals a far more dynamic and interconnected truth. From the Yamnaya sweeping into Europe to the Bantu expansions across Africa, the story of humanity is not one of static purity, but of perpetual motion and mixture. The very concept of an unmixed ancient population is, according to the bones, a historical anomaly. Migration, integration, displacement, and cultural exchange are not exceptions, they are the rule. Our ancestors were travelers, explorers, and neighbors long before borders were drawn. Their remains show that identity has always been more fluid than our modern categories suggest, built through journeys and encounters, not isolation. Perhaps most importantly, this field turns speculation into evidence. For so long, historians and archaeologists had to piece together grand narratives from pottery shards, tool styles, and a handful of ambiguous texts. They could propose that a language spread because of migration, or that a cultural change reflected new people, but it was always an inference, a theory open to debate. Now we have a form of empirical proof. When the isotopes in a Viking-era grave on the Isle of Man show a childhood in Norway, it is not a guess, it is a measurement. When ancient DNA from the Tarim Basin shows a deep local lineage, it settles a century of speculation. This transforms historical mysteries into solvable scientific questions. Legends of the sea peoples, epic poems of migrations, oral histories of long journeys, these are no longer just stories. They are hypotheses that can be tested against the biological record. In many cases, like with the Anglo-Saxon migration into Britain, the science confirms the broad strokes of the written record while adding profound nuance. In others, like with the origins of the Minoans, it completely overturns old assumptions. This is the power of giving voice to the voiceless, of allowing our ancestors to testify on their own behalf through the very substance of their bodies. Ultimately, this is a democratization of history. It ensures that every individual, regardless of their status in life, has the potential to contribute to the grand narrative of our species. A humble farmer buried without grave goods can tell us as much about climatic change and dietary shifts as a king entombed with gold. The story of humanity is no longer reserved for those who could write it down. It is written in the skeleton of a child who moved with their parents, in the teeth of a woman who married into a new community, in the genetic legacy of a trader who settled far from home. This is their history, finally audible, and as we learn to listen more closely, we are not just uncovering where we came from, we are redefining what it means to belong to the endless, interconnected human journey. The true power of this silent archive is its profound objectivity. A king's chronicle is written to glorify, a conqueror's monument to intimidate, but a tooth does not lie. It simply is. It records the water you drank and the landscape you walked, free from propaganda or pride. This biological record fills the vast, echoing silence that stretches for millennia before the first scribe ever put stylus to clay. For the first time, we are not just interpreting the artifacts people left behind, we are listening to the people themselves. Through the precise language of isotopes and the intricate code of ancient DNA, we have gained a dual lens. One zooms in to trace the intimate life journey of a single individual, revealing where they were born, what they ate, and the miles they traveled. The other pulls back to map the epic, population-level migrations that reshaped continents and forged the foundations of our modern world. These are not separate stories, but interconnected scales of human experience, from the personal voyage of a mother moving to a new village to the grand tide of a culture crossing a sea. This is history written not in ink, but in the elemental chemistry of life itself, and this is only the beginning. The technology to read this archive is advancing at a breathtaking pace. Soon, we may be able to extract not just broad dietary patterns, but the specific seasons in which a person moved, or trace the subtle genetic markers of ancient illnesses and immunities, adding even richer detail to these recovered biographies. Each advance turns the page on another chapter of our collective past, so the next time you consider human history, see it not just as a series of dates and battles, but as a deeply interconnected biological story. It is a narrative of constant movement, adaptation, and mixture, written into the very fabric of our bones and teeth. Our shared journey is far more complex and far more united than any single text could ever convey. If you found this exploration into the silent past compelling, be sure to subscribe. We are just beginning to learn how to listen, and there are countless more mysteries waiting to be heard.